All right. As I told you before, in meiosis, what are the two main important phases? Prophase 1 and metaphase 1. Prophase 1, we have seen homologous chromosome overlap, synapsis, synaptomental complex, and crossing over of the chromosomes, and they form this chiasma. And metaphase 1, another important thing happening for both what is happening at prophase 1 and metaphase 1 is to make the genetic variation. So, what is genetic variation? Every individual, not only human, any organisms, uh, they are different to each other. This is getting very less when it comes to the lower class of animals like prokaryotes and other organisms. It is getting less, but they have again different mechanisms. Those organisms also want to make the variation. I think we can better understand variation when we are doing unit 3, natural selection and evolution. There only we learn full version of the variation. But here, what is the importance of meiosis in variation? Then you have to highlight prophase 1, metaphase 1, the changes make the DNA chromosomes different. It's like a shuffling, kind of a shuffling genetic materials to make different. It's a kind of a random process. Random process mean if we look mother and father same in the family, but the siblings, brothers and sisters, they are different. So you can think mother's DNA and father's DNA, they are same, and siblings should get, if they get the same DNA, same genetic material, same genes, why they are different to each other? The major difference happen because of the meiosis. So when the gametes form randomly, they shuffle all the genes and to make a new variations, new combinations of genotypes, and this cause a difference among even with the family members, right? Now, metaphase one, what is happening is like this. Okay, remember mitosis. What is happening in mitosis? Metaphase in mitosis, the chromosome align at the equator, meta plane, uh, the metaplane or metaplane that's in the equator, right? All the chromosome align in the equator. In that case, there is only one chromosomes attached to the spindle. In the middle, there's a one chromosome, and below it, another chromosome like that. Just only one chromosome line up. Now, here is different. The pair of chromosomes, that means homologous chromosomes, line up like this, right? So I draw one, and this is the spindle, right? So these are the centrosome on the poles and as before they are attached to the centromere, kinetochore, spindles, and one centrosome in this pole, the other centrosome in the other pole. It's everything is same. Now you see different. This is a homologous chromosome. They align again at the equator, but this time they are pairs. Now, why it is important? I draw second one, right? Second pair. Right. Now, if you look, what is the difference between first pair and second pair? We can see the white one is on left, the yellow one is on right. Right? In this second homologous chromosome, yellow one is left, white one is right. Now, imagine I'm going to line up all 23 pair of chromosomes like this. So then I have to draw from here to here all these pair of chromosomes. Okay, then how should I draw white and yellow chromosomes? So there's no rule, it's a random. So I can place next one. For example, if I want to draw another one, again I can place yellow one here, and then white one other side. Okay, so I can keep on doing like this for all the chromosomes. There's no rule to move yellow to one side or white to one side. Now, why this is important? Consider the first pair, the homologous chromosome. So, homologous chromosome, it's a very important concept. The chromosome exists in pairs. So, human has 46 chromosome. This 46 chromosome exists as 23 pairs. Why they exist in pairs? I told you these chromosomes are not 100% identical. So they are not 100% identical, but they have similarities. So we can really learn very clearly what is this identical, what is not identical, what is similarities when we learn genetics. So when we learn genetics, we can learn these things. For example, these two chromosomes, one from father, one from mother. Let's say white one is from father. So this is from father, and this one is from father. This one is from father. This one is from mother. This one is from mother. This one is from mother. Now, you don't have if good knowledge in genetics. It is very difficult to understand at this point why these homologous chromosomes exist in pairs. I can tell you something just to understand only at this point, but later, uh, when you learn genetic, you get the complete picture of this. Now, what is this chromosome? The chromosomes are DNA molecules. They compress DNA molecule. What is inside the DNA? So, inside the DNA, there are nucleotides. What is the significance of nucleotide? Nucleotide, order of nucleotide make a gene. Order of nucleotide make a genes. So then the chromosomes separate into genes. What is gene? Gene is a specific sequence of DNA which can make a polypeptide or protein or a special character, right? Let's see one example just to understand. Now, father side, the first chromosome, there is a gene. This gene responsible for the hair color. Here, it's the one I'm marking. This region is the DNA responsible for hair color. Now, mother side, the same region, it is also responsible for the hair color. Now you see similarity. What is similarity? Ah, the chromosome number one is responsible for hair color of a person. This is example, right? I'm not telling about the exact chromosome and this is the right one, just to only learning purpose. Chromosome number one, the father and mother, this place has a gene which make the hair color. Now, what is different? Different is the father gene make a black hair color, mother can make a brown hair color. Now you see the difference. They are not identical, but they have many similarities, but they are different. That's why we call homologous chromosome. This likewise, all the genes are located and they are responsible for same kind of feature, but they are different features. Now, this alignment, it's a random, right? It's a completely random. So this is also important for the variation. You can now understand it's a mixing mother and father genes by aligning to one side and by aligning to one side and they are differently. Okay, this is father side, this, mother side, this, like that. It's again, we are shuffling the information or characters or proteins or genes, whatever we are doing, father side and mother side we are mixing. So then the person who is getting this, I mean, at the end when it's separate, I make a line just to a little bit understanding. So if you align all the 46 like this, 23 pairs, then when it make a cell, this side chromosome is going to this cell, and this side chromosome is going to this cell. So then two cells are different because they have a different genes from mothers and father. It's like a mixing. So Metabase 1 resource book doesn't use this word, and also Campbell book doesn't use this word random assortment or random assortment or independent assortment. This word random assortment and independent assortment, they are very important words. Sometimes we don't see in resource book because of the Campbell, because Campbell book doesn't use this word, but they are standard word. Now I see the difference between the resource book and in the paper if we have something not in the resource book, because these are standard word and the examiner can use the standard word without thinking, and I mean he doesn't know, okay, we never use this book in the uh, resource book and we never use this book in the Campbell, so we shouldn't use that word because student doesn't know. We should use same word uh, which use in the resource book, but he might forgotten this one because other words sometimes are standard word. Now random assortment and independent assortment are very standard word. Almost all textbook they use this word in Metabase. For example, I will show you another word, photosynthesis. The Campbell book and resource book use water splitting. They use the word water splitting, but the standard word is photolysis. And you will find in exam paper, the recent new papers, they use photolysis, not water splitting. There are minor things are there. So if you want to learn a little bit more or additional things, it is only very little things, not much. Right? It's not much because still for this kind of a small thing, we cannot neglect, we cannot downgrade. The resource book is not a one to refer. If you look, I think almost all teachers they will say yes. You have this resource book. It is the one you had to study, not the other books, right? So Campbell book is there, but I'm saying don't use Campbell book for studies. If you want diagrams, pictures, other things, you can look. But study purpose, only the resource book, right? There are minor things which is not big things to talk about. We can correct and we can learn, right? So remember now random assortment, organization of chromosomes in the middle equator, and that's completely random. After that, it's a mother and father chromosome mixed up on one side and also other side. Then when it's separate, it also brings the differences. It's not the same because when the process is random, then the cells make they are different. They get a different numbers. There are different numbers from mother and father. Like the chromosomes are same, but they get different type of chromosomes because of, for example, if it is happening second time, so next cells are making second pair of chromosomes. Yellow one going to the right, white one go to the left. So then that's a difference. So they can make differences at each time. Okay, so clear again, keep in mind the variations of living being are made by propase 1 and metapase 1 changes. So that's the most important changes, bring the variations, differences among the organisms. So I just highlighted this independent assortment, not in resource book, but that's a very standard word. But now we understand what is happening at the metabase. Right now, both meiosis, propase 1 and metapase 1, the changes make human, every individual different to each other. So that is the main contribution for this variation coming from the changes happened at propase 1 and metapase 1. But there are other things, but this is the major two factors variation comes to human. Okay, then what is going to happen anaphase? Well, it is same as before in mitosis. That means they can split just surely one. The homologous chromosomes separate. Homologous chromosomes separate, and one the left side chromosome is going to the left side, right side chromosome is going to the right side. So now homologous chromosomes separate, right? You should also understand now when we say meiosis and also mitosis, what is mitosis? It make genetically identical cells. So mitosis has cells has 46 chromosomes. One when it divide to make two cells, each cell has again 46 chromosome, 46 and 46. Now when we always see full set of chromosome, we call it diploid. Diploid cell has full set of chromosome. If the chromosome number become half from the original, we call haploid, right? Uh, Also, we should understand another important thing. If we go, let's go number wise. What is number wise? Like this. Now, 46 chromosomes should be duplicated to make 92. So it's 92, and after that, 92 going to one cell, 46 and 46. So then we can understand, yes, it's again same. Now let's count the chromosome number in meiosis case. So what happened in meiosis? Meiosis again, 46 duplicated become 92, right? So where is the duplicated one? I think I showed you many times. Again, I'm showing. So duplicated one, let's say this. We call it sister chromatids, right? So that's a sister chromatid. Now, how many we can see in this side? 23. 23 chromosomes. Now, 23 chromosomes, duplicated one. Duplicated one mean it is again 46. So this side has chromosome number 46 and this side has a 46. Now, meiosis 1, when it makes two cells, one cell has 46, other cell has 46. So it's a meiosis 1, again 46 and 46. But we are not concerned about the number, right? We are not concerned about the number. At this case, end of meiosis 1, we call it is reduction division. Chromosomes become half. Chromosome become half. Now, bit complicated, right? You see 46, but now it's saying chromosome become half. At the end of meiosis 1, let's say, I draw like this. Start of meiosis, 1, 92, 46, 46. And after that, it is starting next stage, meiosis 2. So meiosis 2, this cell become 23, 23, and this 23, 23. Right. Now, if it is, this is duplicated one from 46, right? Now, if you look at this 23, it is half a number of chromosome. Why? Because there is a human has 46 chromosome, and now it has 23 chromosomes. But we consider the real reduction division, the reduced chromosome, happen at this place. We consider this place. The reduction happened at this place. Why? Because it's a 46. How we can say it's a reduction division? Now look at these chromosomes, homologous chromosome. So we consider the separation of homologous chromosome, because if the cell has 46 chromosomes, it is full. But homologous chromosome separate mean half of the information gone. It is duplicated here, but duplicated mean this one and this one is same. This one is same. So information, we are concerned not the number, we are considering the information, genetic information become half. Because one cell lost this chromosome. Homologous chromosome, I told you homologous chromosome is the pair. It exists in pairs. They have information. This has different information, this has different information. If both in one cell, they have full information. But if we separate, then half of the information gone. So it is not the number, but we go for quality wise. What is the quality wise? Genetic information become half at the end of meiosis 1. So it's called reduction division. So anapase, it is separating. That means microtubules become shorter. They pull the chromosomes to the opposite pole. That's a centrosome, and then chromosomes are moving to these opposite poles. So if you are still confused, you can look first number wise. Okay, this is happening. Then if you take 46, actually it has 23, because it's a duplicated one. Because when this cell get a chromosome, the yellow color one and the yellow color one both are same because the duplicated one. But other side, other cell, it get a homologous pair. That means I make a red color like this. If the yellow and red in the same cell, it has all information. But here only one cell get yellow, other cell get red. That means genetic information getting half. Because of that, we consider reduction division, or the chromosomes number, the chromosomes information is getting half at the end of meiosis 1, right? And after that, uh, most of the textbook, they mention meiosis 2 is same as mitosis. Meiosis 2 is same as mitosis. Every event, it is exactly same as mitosis. That's why I told you, don't be complicated, make it confused with cell division. It is very easy to understand. Only a few stages you need to remember well. Rest of the thing you can make it based on the other your knowledge you have. So few things to remember. Rest of the things, like for example, if you remember events in mitosis, it's the same here. Same thing happened in meiosis 1. Sorry, meiosis 2. So prophase 1 and metaphase 1, you remember well. And also try to understand, end of the meiosis, the genetic information become half to the one cell, half to one cell. Then it's a really divide the information into two cells. So reduction happen at the end of meiosis 1, right? Okay, next, telophase 1. So telophase 1, homologous chromosomes, they are moving to this side. Other homologous chromosomes, they are going to the other side. If you look at the telophase in mitosis, you see only a one. Here so still we have the duplicated one. And what are the other things? Other things same as mitosis. That means nuclear envelope start to form, and nucleolus started to appear, and chromosomes become a little bit invisible like this. So there is again consideration: prophase 1, metaphase 1, anaphase 1, telophase 1. After completion of these four, whether the cell stay a little bit longer, or cell is waiting to go to the meiosis 2, or immediately go to the meiosis 2. This is depend on the cell type, and we are not very much interested all these events in every events. So we only focus on the most important things, and then we continue for the rest of the events like just it happened. It is different from cell to cells. Some cells, they stop at certain stage, and there is something stimulus happen, then they continue. You will see this one very clearly in the human reproduction. the egg cell or ovum, when it release, it is in a different stage of meiosis. If the sperm cell fertilize, then they complete rest of the events. Something like that, right? So we don't want to consider too much, but we go uh, to the events, and it is always wise that you focus the most important things. Unnecessary thing, it, you can learn. There's nothing called unnecessary, but the problem is, uh, at the end of the day, you have a knowledge, you have a very good knowledge, but if you can't pass the exam, it is a different story. Because recently I had a student, uh, he came, sir, we made a mistake. We learn everything from the Campbell book. So A to Z Campbell book, they learn, didn't get the correct guidelines. It is too much. It's very hard to focus, because the exams, like this exam is different. Let's say you go to medical faculty, you are doing exam at medical faculty, then what you tell in oral exam, or what you write, that's your knowledge. They don't check whether this is correct with the marking scheme. Did he write this marking scheme point? Did he write marking scheme point? Now they go generally how judge your knowledge, not one to one. They are not checking with the marking scheme. But until A level, especially A level exam, the challenge is with right to the marking scheme. University exams, examiner will not check your answer with the marking scheme. They check your knowledge, how wise you are, whether you are aware of everything, you know this area. But let's say uh, you write something very advancing in the answer. It is right, and it's a very advanced thing you wrote, which is correct, but marking scheme not there. They don't want to bother to give marks for this one. But you do the same thing at the university exam. The professor will very happy. You are updated with knowledge. Good. It's very good. You know these things. So it's a different story. That's why we have to learn according to the things we have to learn, right? Many students, they made a mistake. Too much knowledge is not making marks higher. So only the resource book we have, that we have to study, right? Okay, telophase one, same as the mitosis, and then next after that meiosis two, right? So meiosis two, again starting with prophase two, metaphase two, anaphase two, telophase two. This is same as mitosis. Events are same as mitosis. I repeat, events are same as mitosis. So let's revise quickly. Prophase 1, prophase 2, what is happening? We have seen new chromosome condense visible, the nuclear envelope disintegrate, nucleolus disintegrate, attach the chromosome attached to kinetochore, and the chromosomes are more visible at the end, at the prometaphase. And after that, a metaphase 2. Then all chromosome align the equator like this. Then anaphase, it breaks from the centromere and move to the opposite poles. And telophase 2, this should be telophase. So telophase 2 again, a nuclear envelope formed, nucleolus formed, chromosomes are not visible, and spindle becomes shorter, are not visible. Right. Last, for any cytokinesis for mitosis and meiosis, the cytokinesis in animal cells, the cell start to divide into two, like this, and this is called cleavage of furrow. Okay, now I will ask you a question. Uh, what is the role of the filaments of a cell responsible to make the cleavage furrow? Which filament? You learn it. Okay. Yes. Can you remember? Chitsara. Very good. Microfilaments of actin. Now, this is the knowledge you need, right? So, what we done in the past, go check microtubules, microfilaments or actin or intermediate filaments. Under that, we listed the functions. Now, if you can keep up from the beginning, your run rate higher, I think exam is nothing, right? So consider this is like a 2020 match. Don't wait until the last overs to come. Score from the first overs. Start to score from the first overs. Uh, every person, every human, we have some weakness we are forgetting. It's happening to everyone. No one in this world they have the memory always same if they don't update. So we have to update our our memories because I will tell you something very important for your studies. Uh, there is research done in the past, and many research, and scientists build up something called forgetting curve. So all humans, we have this forgetting curve, and they proposed for student if they learn something today within 24 hours. I mean, you can Google forgetting curve, you get this curve. Within 24 hours, if you revise your lesson you learned today, they recommend to revise within 24 hours. And after that, 3 days, after that, 7 days, like that, you have to keep on revising like with the gaps, increasing gaps. So when you revise with the increasing gap, the retention is very high. If you don't revise for a week, let's say today you learn this lesson, now you are coming next week and try to recall what you learned. They say 80% drained, 80% knowledge drained. Check it for this kind of exam for you. It's very important. Uh, Google search forgetting curve. You will see it, and the guidelines are very important for you. How you have to revise the lessons, like monthly, weekly, or hourly. How you have to plan your revision lessons. It is not close to the exam. Keep on right now. Uh, I mean, you don't want to stay 24 hours. Just if you have a good plan, you can manage it. Yes, that is important. Microfilaments, they make this string around it, and also the Golgi-like things in the middle. They help to make these separations.